In a previous video, I mentioned that there were 20 different types of amino acids. Now, what I want to show now is in this table is that although there are different types of amino acids, they all share a very common structure, and that structure we already looked at. So let's just concentrate on the first amino acid here, which is known to be glycine. Okay, so let's just concentrate on that one for just a moment, and then we'll see that once we understand this, we can move on to the other ones without too much problems. So we know that one thing that is common to all amino acids is to have something that we call here a central carbon, or also known as the alpha carbon. So this alpha carbon is right here in the middle. Connected to this alpha carbon, we have our carboxylic acid right here, and also we have an amine group right here. And we know that those two groups here are very important when we want to link many different amino acids together and form a peptide bond. Now, another part of these amino acids that is common is to have a nitrogen right here. So basically, when you look at all of the other amino acids, right? So we can look at all of these ones here. We know that this is always common to every single one of them. So just look at those with the few exceptions, but most of them have basically the same kind of structure. The difference between the different amino acids is based on what we call the R group. So what is this R group? Well, let's just go back to our carbon alpha right here. So we have the alpha carbon right there. And then I'm going to choose another color. I'm going to choose, say, green for this. And I'm going to show that we have different R groups. So we have our basic structure as we demonstrated. But also we have, you see right here for the amino acid glycine, we have a group here that is a single atom of hydrogen right there. So in the case of glycine, the R group is equal to H right here. However, when we go to other types of amino acids, we keep the same basic structure, right? But in the case of alanine here, the R group is what changes. And in this case here, we have a carbon with 3 hydrogens connected to it. If the R is equal to this group right here, then we have alanine. If the R group is this group that we find right here, then we have valine, and so on and so forth. So you see that the R group can become more complex as we go, right? So just look here at the bottom when we look at tyrosine. Well, tyrosine has a cycle in it just the way phenylalanine has one, and then tryptophan has one. So those are basically the most complex amino acids that we have. And the only difference between tyrosine, phenylalanine, alanine, glycine, valine, and all that is the R group. Now I say, well, the only difference, as if it was that simple, right? Well, it is not. There is a small difference, but that difference is not that small when we look at those groups, right? The difference in chemical structure of these groups is huge. When you compare, say, glycine with tyrosine at the bottom, we know that there is a big difference there. And of course, the properties of these amino acids, namely glycine and tyrosine, is going to be completely different when they're linked together in a peptide bond within a protein. So before we move on, I just want to remind you of something simple but super important. So we have 2 amino acids here. So let's say that's number 1, and then we have number 2 right there, right? Those 2 amino acids can be united together to form something that we call a peptide bond right here. So we see that a peptide bond will be formed between the carboxylic acid right here and the amine group right there. And this will lead to the formation of a water molecule right here. Remember that this is a reaction of condensation, right? We don't see the water coming out in this reaction here, but we know that this part here will be eliminated as a water molecule. And then we will form this peptide bond that is written and shown right at this spot here. Now, one thing that I wanted to connect with the previous part of this video is that we have the R groups here, R and R. Remember that R group is only something that we put on paper when we don't really want to deal with the structure that's found here. Basically, a peptide bond can be established between any of the 20 types of amino acids, even though the first amino acid can be the same as the second one. And in this case here, we know, we look at the R group for the first one and the R group for the second one, we see that both of them are exactly the same one. And I'm going to question you here, do you remember which amino acid it is when the R group is equal to H, to hydrogen? I'm not going to answer that question. I'm just going to ask you to look at the previous part of the video, and I think you're going to have the answer right there. So this figure illustrates a bunch of amino acids that are connected together via a peptide bond. So the peptide bond would happen between every single one of those amino acids at this spot right here, right? So we have glycine, serine, histidine, leucine, etc. And every single one of those amino acids are connected together. They're linked via a covalent bond, and that bond is known to be a peptide bond. The reason why I'm showing this figure to you is to remind you that when we have a bunch of amino acids connected like this with one another, this is known to be the primary structure of a protein. So one thing that I need to mention here is that you will hear me talk about different concepts here. So the first and basic structure of any protein, the building blocks of any protein, is the amino acids. We've seen that there's 20 of them. Those amino acids will interact with one another, they'll be connected with one another, and they will form small chains. Those small chains are known to be peptides. When a chain contains only maybe 50 amino acids or less, we call that a peptide. As the chain gets bigger and bigger, longer and longer, that peptide is now called a protein. Now, what we're looking at here is the primary structure of a protein or a peptide, depending on the final length of this. Doesn't matter if it's a protein, doesn't matter if it's a peptide. The sequence of amino acids, the ones that are linked with one another, is known to be the primary structure of a peptide or the primary structure of a protein. So as we get things a little bit more and more complex as we go, as we get a longer and longer primary structure, we know that the different amino acids in the primary structure, so you see we have one here, one here, one here, one here, and as we go on, as the chain gets longer and longer, these amino acids will have a tendency to associate with one another in different ways. And the different ways these amino acids will interact with one another is based on the R groups of every single one of those amino acids here. So you see every single one of those amino acids have R groups right here, right? And depending on those R groups, the amino acids themselves, the different balls that we see here, the different amino acids, will shift in space in one way or another. And because of that, these amino acids, when they're close to one another, will have a tendency to form different types of structures. And there's two main different types of structures that are available or that can form. And those are known to be alpha helix and we see here there's this corkscrew structure right here, that's a helix right there, and we also have something it's called the beta sheet or beta pleated sheet, as we see here. So what we see here is every time you see this part here, this is where the peptide bond is, right? So the peptide bond is right here. So we have one amino acid number 1 here, amino acid number 2 here, amino acid number 3 here, and those amino acids are linked together and form those structure, either the alpha helix or the beta sheet as shown right here. So the model that is shown here is known to be a peptide hormone called insulin, and insulin is used by all of the cells of your body to get glucose from the bloodstream. Now I'm not going to get into this right now, not at the moment, but I just want to show that even though this is a peptide hormone, this is a pretty small protein, a small assembly of different amino acids, and we see all of those amino acids here, right? So you see every time you see a blue region right here, all of those blue regions are known to be the amine groups. And then when you see those groups right here with the red on both sides, we see them everywhere. This is a carboxylic acid group. So I can rotate this model in space and we can look at it and see what it looks like. So we see that it's kind of bulky. We can kind of guess the primary structure of this model right here. We know that the amino acids are linked with one another. It's very difficult though to make out where's the beginning and where's the end of this structure. However, if I show something that's called the ribbon structure, we can see that we can start to see the structure of this hormone, this peptide hormone that is insulin. And you can tell right here that we have the alpha helix right there, right? So we see that those residues, so those amino acids that we have in this region here, are actually forming these alpha helix, and if I turn them the right way, we can see that these helices form a cylinder, right? We have one here, and we have another one right there. It's very easy to see. So those are the alpha helix of this protein, or this peptide hormone. Now, if we simplify things a little bit more, we see that we have this chain here, this peptide chain. We can see in purple the color of the alpha helix. The portions in white are portions of the peptide that are not really forming any particular secondary shape. So the secondary shapes here, secondary structure of this is the alpha helix that we have here, and we have another one here, another one here. So we can see that there's 3 portions of this structure that can form a alpha helix. Now what we have here is another structure that's way more complex than insulin. So this is a protein. This is not a peptide. It is a full-blown protein, quite big, and it's called carboxypeptidase. It's a digestive enzyme. We'll see what enzymes are and what it means a little bit later, but right now I just wanted to show the secondary structures. So we see that in purple again we have our alpha helices right here, and we see there's many of them. But what I wanted to illustrate here is that within these structures of alpha helices all over the place, we have in blue this portion that we see here, and this portion is the beta sheets that I talked about earlier, right? So when we see those beta sheets, although when we look at the alpha helices, we see they're form cylinders. When we look at beta sheets, see why we say it's forming a sheet? It's forming a sheet because the structure is actually, it's not so much like in 3 dimensions. It's more like a 2D organization. So we see that here, right? We look at it and there's not a lot of depth to this structure right here. So we know that beta sheets are more organized in parallel strands like we see here, whereas the alpha helices are organized and they form cylinders on top of each other. And when we change the way we look with the different tools I have on this website, and I'll give you the address of this website. You can play with this protein as much as you want. It's quite interesting to do. Now, when we change the organization and the visualization of this protein, we can see now that the alpha helices are depicted as cylinders. That's pretty common. You see this in publications all the time. We have the white parts right here. We know that these white parts have no real secondary structure to them. And then we see in blue right here, it's much easier to see now that this beta sheet is mostly bidimensional, as we can see right here. It's organized in space for sure, but it's more on a plane, if I can say it like that. So hopefully by now you have a clear understanding of what a primary structure is and what a secondary structure is. So we have our alpha helices here. There's many of them. And then right here we have our beta sheet at this point. And this beta and the alpha helices are known to be secondary structures. Now, when we look a little bit deeper into structures of proteins, we know that there is another type of structure that's known as tertiary structure. And the tertiary structure is the interaction of each of those different secondary structures with one another. So here's what I want you to understand here is this. These alpha helices are held together like this because the amino acids found in them interact with one another, as we've seen before. The interaction between the amino acids found in these alpha helix or helices, the interaction is known to be hydrogen bonds, right? So H bonds. And the same thing is found for the portions of the beta sheet here. The interaction between every strand of these beta sheets here are also held together by hydrogen bonds. So when we look at the tertiary structures of the proteins, we're looking now at the interaction between different parts of the secondary structure and how those different parts interact with one another. Because they do interact with one another. They're held together in a certain way. And we know that certain parts of this protein here, there's different ways proteins interact with one another. And all of these interactions hold together this protein and provide to this protein a very, very specific structure. That specific structure that is the result of the interaction of the different elements of the secondary structure are known to be the tertiary structure. So this is what we have here. We have all of this bulky protein right here, and what we see here is the result of the interaction of all the parts of the secondary structure forming this little ball, and this is known as a tertiary structure. Now, one thing that is very important for you to understand is this. This is carboxypeptidase. It's a type of protein. And carboxypeptidase will always produce the same shape. It doesn't matter in which individual it's made, it's always forming the same shape. The beta sheet will always be where it is. All of the alpha helices will be around this beta sheet in the same way, interacting with this beta sheet in the same way it is. Remember that the forces uniting all of these amino acids together are physical and chemical forces, and whenever you have the same sequence of amino acids in the primary structure, you have the same types of interaction in the secondary structure, which will provide the same shape of protein in the tertiary structure that we see here. Last thing about tertiary structure is that do not forget that structure equals function. So say that in a laboratory, I decide that I can eliminate this alpha helix right here. So let's say that there's a way for me to strip this protein from this alpha helix right here so it completely disappears. Well, I can guarantee you one thing, is that the shape, the overall shape of this protein here will be completely changed because we have such a big chunk missing. And I say a big chunk, you don't even need a big chunk you don't even need to remove a big chunk to alter or change the function of a protein. Sometimes all you need to do is change a tiny amino acid right here, and this would change the function of this protein absolutely completely. So don't forget structure and function. Now for the final part of my protein structure talk, I want to show you this wonderful molecule here, and this molecule here is known to be hemoglobin. So when I look at this molecule here, I can turn it in space. We can tell that it's pretty, bulky, pretty big. There's no beta sheet to this structure. There's only alpha helices and a bunch of threads in between that link the alpha helices with one another. Now the interesting part comes when we color the different chains of this protein assembly, I should say. So now if we spin this in space again, we see that we have four main colors, right? We have the red one, we have blue, green, and sort of yellow at the back right there. So when we get to these proteins here, these proportions here, I want you to focus on, say, the green part right here. So I'm going to rotate this piece, and I want you to really pay attention to the green part of this assembly right here. So let's look at that. So if we look at this green part here, right, just look at it, and then notice, hopefully you'll be able to notice one thing. And I'm going to tell you. So it's quite obvious right here when I position this molecule this way. So what I want to show here is that this green part of the hemoglobin is not connected to any other part. You see, it's kind of like free floating. It's not connected in any way by any structure, any primary structure, I should say. It's not connected to the other part of hemoglobin. Now, if we look at another part of hemoglobin, let's now focus, say, on the blue part. So I'm going to bring the blue part forward right here, right? So let's just look at it. Let's bring it forward. And hopefully what you will see, and what I'm attempting to show here, is that the same thing happens with this part. The same thing happens. This part of hemoglobin right here is not connected in primary structure with any other part of hemoglobin. And we know this is true for the other parts as well. So basically, what we see here, the 4 different colors of hemoglobin here, right? The 4 different parts with different colors, they are not connected via the primary structure. So those 4 parts are known to be subunits. And those subunits are interacting together via physical and chemical forces, are holding this whole thing together. So basically what I want to show here is that every single color is a tertiary structure that's maintained together. And all of these subunits are interacting with one another. And when we have different subunits interacting with one another, we say this is a quaternary structure. Now, not all proteins have quaternary structures. All proteins have a primary, secondary, and tertiary structure. But some proteins, and only those that have subunits, will be able to form quaternary structure. And that is the case in hemoglobin. So hemoglobin is an assembly of different subunits. 2 of those subunits are absolutely identical. So we have subunit alpha 1, and then we have subunit alpha 2. And those 2 subunits here, alpha and alpha, are exactly the same. On the other side, we have 2 other subunits that are absolutely identical as well. We have beta 1, and then we have subunit beta 2. So those 2 beta subunits are exactly the same. Now, when we look at those different subunits, so let me change the color. So we have a subunit right here. So let's look at that. So we have our alpha subunit right here in a 1 color. And then we have our other one that's right here. This is our other subunit. Behind that, we have another subunit right here. And then at the back here, we have another subunit. So all of those subunits are united and they are interacting with one another in what we call a quaternary structure.